Bones don't lie. But for decades, the bones buried across Southeast Asia have been silent, not because they had nothing to say, but because something in the ground itself was working to erase their voice before anyone could hear it. Now, using techniques that did not exist 10 years ago, scientists have pulled fragments of genetic code out of skeletons that were never supposed to survive long enough to be read. And what came out of that code is not a footnote to human history. It is a challenge to almost everything we thought we understood about where we came from, how we spread across the earth, and who was already here when we arrived. Somewhere in the humid caves of Indonesia, Laos, and southern China, the official story of human origins just cracked. And once you see the crack, you cannot unsee it. For most of the 20th century, Southeast Asia was treated as a blank space on the map of human genetics. Not because nothing happened there, but because almost nothing survived. The heat, the humidity, the acidic soil, the relentless biological decay of the tropics, all of it worked together like a machine built to destroy evidence. Ancient DNA needs cold, dry, stable conditions to survive for tens of thousands of years. Siberia has that. The Arctic has that. Europe has that. Southeast Asia has almost none of it. So while European skeletons were giving up secret after secret, Southeast Asian remains stayed locked, decayed, unreadable. And the region that may have been the single most important crossroads in the story of our species became the quietest one. That silence was mistaken, again and again, for absence. Scientists assumed less happened there simply because less could be recovered. But here's where it gets worse, or better, depending on how you look at it. Silence is not the same as emptiness, and when a handful of extraordinary caves finally gave up their secrets, the silence broke all at once. The first tremor came from a cave in South Sulawesi, Indonesia, called Liang Panenge. Buried there for thousands of years was the partial skeleton of a young woman. Skull fragments and pelvis, nothing more, but enough. Enough for researchers to reach into the dense petrous bone at the base of her skull, the single densest bone in the human body, the place where genetic material hides the longest before the tropics can destroy it. What they pulled out of that bone should not have been possible. Her DNA revealed something that did not match any population that had ever been sequenced before. She carried ancestry more closely related to modern Papuans and indigenous Australians than to any mainland East Asian population alive today. Read that again. A woman buried in Indonesia carried genetic fingerprints pointing toward Australia and New Guinea, not toward the Asian mainland right next door. That single fact rewired the map. It meant she belonged to a previously unknown, deeply distinct lineage of humans, one that geneticists now believe split off from other populations around 37,000 years ago, almost exactly the same window in which Papuans and Aboriginal Australians are estimated to have diverged from each other. In other words, her ancestors were present, embedded, and already diversifying in island Southeast Asia, while the rest of the world was still working out its own migrations. But that wasn't even the most explosive part of what her bones revealed. Woven into her genome was something that should not have been there at all if the textbook story were true. A measurable trace of Denisovan ancestry. Denisovans, the ghost population. The hominins we know almost entirely from a handful of finger bones and teeth found in a Siberian cave, and yet whose genetic fingerprint turns up thousands of miles away, in the islands of the Pacific and Southeast Asia, in populations that supposedly should have had no contact with them at all. This woman lived roughly 7,300 years ago. Her Denisovan ancestry means that interbreeding between modern humans and this archaic population wasn't a brief, isolated encounter somewhere in Central Asia thousands of generations ago. And then, nothing. It means that Denisovan genetic material was still circulating, still being passed down, still shaping the biology of real, living communities in island Southeast Asia, deep into what we consider recent prehistory. This is not chaos. This is a signature. And once you understand what a signature like this means, you start asking the question that should terrify every tidy textbook diagram of human migration. How many other signatures are still buried out there, waiting? Because Sulawesi wasn't an isolated anomaly. It was a warning shot. Move north, across the water, into the limestone cave systems of Laos, and you find Tampaling, a site that has quietly been rewriting the timeline of when modern humans first pushed into mainland Southeast Asia. For years, archaeologists working microscopic layers of sediment inside that cave have been pulling out evidence, not just bones, but the residue of fire, of habitation, of humans actively living and working inside that space far earlier than conventional models predicted. Some of the human remains recovered from Tampaling have been dated to a staggering range, with the earliest evidence pushing close to 70,000 years of possible habitation and skeletal material, confidently dated between 46,000 and 70,000 years old. Now here's the part that most people watching documentaries never get told. Those bones are so old and the tropical conditions so hostile that direct nuclear DNA extraction from the skeletons themselves has repeatedly failed. The genetic material is too degraded. It has been baked and dissolved by tens of thousands of years of heat and moisture. And yet, scientists are not giving up. They have started extracting DNA not from the bones directly, but from the sediment surrounding them, the literal dirt the body's decomposed into, which still carries fragments of human genetic signal locked inside microscopic particles. Think about what that means. We are now at the stage where scientists can potentially read the genetic ghost of a human being from the soil they were buried in, even when the skeleton itself refuses to give up its secrets. That is not archaeology anymore. That is closer to something out of a thriller, pulling a signal out of decayed silence. What most people are not seeing is why this matters beyond the individual skeletons. Tampa Ling and sites like it are proof that modern humans were pushing into mainland Southeast Asia far earlier, and far more persistently than the simplified single wave out of Africa narrative ever accounted for. According to the broader body of research building around these tropical Asian sites, the peopling of this region wasn't one clean migration event. It was messy, overlapping. Multiple waves, multiple routes, some populations disappearing entirely, others merging, others staying isolated in pockets for tens of thousands of years before finally showing up in a genetic sample that finally, after everything, survived long enough to be sequenced. And each time a new sample survives, the old model takes another hit. This is the part that should unsettle you. Think about what that means. There is a gene, GLI3, that specifically controls how wide your nostrils are. There's another gene, RUNX2, that controls how wide your nose bridge sits. There is DCHS2, sitting on chromosome 4, that determines how pointed or flat your nose tip is and how far your nose projects from your face. And then there's PAX1, influencing the shape of your nostrils. Each of these genes contains variants, slightly different versions, that produce slightly different outcomes. The combination of variants you inherited determines, with remarkable specificity, the nose that appears on your face. This is not vague biological inheritance. This is a blueprint, a detailed architectural plan executed in the womb by molecular machinery that has been refined across hundreds of thousands of years of human evolution. GLI3, DCHS2, and RUNX2 are known to show strong signals of recent selection in modern humans compared to archaic humans, such as Neanderthals and Denisovans, with GLI3 in particular undergoing rapid evolution. Rapid. That is a word that rarely appears in evolutionary biology because evolution on the genetic level is usually an imperceptibly slow process. When a gene shows a rapid rate of change, it means selection pressure was strong. It means the environment was powerfully rewarding certain versions of this gene and powerfully punishing others. It means nose shape was not trivial to your ancestors' survival. It was significant enough for natural selection to actively accelerate the evolution of the very genes that build it. And this is where the story takes its most astonishing turn. Because in 2023, a team of researchers, led by scientists from University College London, published a study in the journal Communications Biology that revealed something that nobody expected. Something that connects your nose, not just to your ancestors in Africa or Asia or Europe, but to an entirely different species of human that vanished from the Earth approximately 40,000 years ago. Humans inherited genetic material from Neanderthals that affects the shape of our noses. A study in communications biology found that a particular gene, which leads to a taller nose measured from top to bottom, may have been the product of natural selection as ancient humans adapted to colder climates after leaving Africa. The Neanderthals, the species we were taught to think of as primitive cousins who died out and left nothing behind. Except they did leave something behind, they left it inside us. According to informed sources in the field of paleogenomics, the discovery that modern humans carry Neanderthal LDNA is not new. It has been known since the first Neanderthal genome was sequenced in 2010, a breakthrough that earned Svante Pääbo the Nobel Prize in Physiology or Medicine in 2022. But what the 2023 study revealed was that this ancient inherited DNA specifically influences the structure of the human nose. In one particular genome region called ATF3, many people in the study with Native American ancestry had genetic material in this gene that was inherited from the Neanderthals, contributing to increased nasal height. This gene region has signs of natural selection, suggesting that it conferred an advantage for those carrying the genetic material. Natural selection retained it. That means it was useful. That means when ancient Homo sapiens migrated out of Africa into the colder, drier environments of Eurasia and eventually the Americas, the Neanderthal variant in the ATF3 region provided a survival advantage precise enough for selection to keep it in the gene pool rather than allowing it to fade. The researchers working on this study examined more than 6,000 Latin Americans, a population that is genetically admixed, meaning they carry ancestry from multiple ancient populations. streams including indigenous Americans, Europeans, and West Africans. This admixture made them an extraordinary scientific lens, because in a mixed population, you can observe the effects of individual genetic variants against a rich and varied background. And what the researchers found was that the chromosome region 1q32.3, the region containing the Neanderthal introgressed DNA, was the single strongest genetic signal for nasal height in the entire study. The novel region in 1q32.3 shows introgression from Neanderthals, and the introgressed tract increases nasal height, consistent with the differentiation between Neanderthals and modern humans. Neanderthals were known from skull fossils to have markedly taller noses than modern humans. And now science has confirmed that some modern humans still carry a piece of that Neanderthal nasal architecture dormant in their DNA, passed forward through tens of thousands of years of interbreeding, migration, and survival. But here is where it gets worse, or rather, where the story becomes even more layered. Because the question that naturally emerges from all of this is, why did ancient Homo sapiens interbreed with Neanderthals at all? And the answer, according to the current understanding in paleogenomics, is simply proximity and time. When modern humans left Africa approximately 60,000 to 70,000 years ago and entered the territories where Neanderthals had been living for hundreds of thousands of years, they did not wage immediate and total war. They lived alongside them, they encountered them, and sometimes they reproduced with them. The genetic traces of those encounters are still present in the genomes of most non-African modern humans today, typically representing somewhere between 1 and 4% of total DNA. That number sounds small, but when you consider what those scattered percentages contain, immune system variants, metabolic adaptations, cold weather responses, and yes, nasal architecture, they stop sounding small, they start sounding consequential. What most people are not seeing is that this is not just a story about nose shape, this is a story about survival strategy. When modern humans moved into new, harsher climates, they had two available mechanisms for adaptation. They could wait for random mutations to slowly evolve new traits across thousands of generations, or they could acquire already evolved traits from a species that had been living in those harsh environments for far longer. Neanderthals had occupied cold Eurasia for approximately 300,000 to 400,000 years before modern humans arrived. They had already solved, at the genetic level, many of the problems that Homo sapiens was just beginning to face. The interbreeding that occurred was not just an accident of cohabitation. From an evolutionary standpoint, it was a shortcut. Modern humans borrowed evolutionary solutions from Neanderthals that would have taken thousands of years to develop independently, and the nose was one of those solutions. It has long been speculated that the shape of our noses is determined by natural selection, as our noses can help us regulate the temperature and humidity of the air we breathe in, and different shaped noses may be better suited to different climates that our ancestors lived in. The Neanderthal variant that increases nasal height was likely adaptive in cold, dry climates, precisely because a taller nasal passage increases the internal surface area available to warm and humidify incoming air. A broader,